Good morning, students. In last class, we have studied regarding the classification of fungi. Today, we will study regarding the virus and lichens. Some points we have already knows about the viruses that they are the connecting link between the living and non-living. They are non-living outside the body, but when they enter inside the living cells, they will be get active and act as a living body. Okay, and we know the reason behind it because the viruses has lack the enzyme system. They do not have any type of the enzymes. That's why they lack the uh, life outside the living cell. But discovery is continuously going on again and again, and the prior discoveries has been found incorrect. As earlier we said, enzyme system is absent, but nowadays we have found that certain enzymes are present in viruses. So how the virus has been discovered? Actually, uh, Ivanovsky, he made the pulp of tobacco pulp, and filter out it by Chamberlain paper. Okay, it is very thin that only some particles can go through it, rest of the ground. But what he got that out of this chamber, what fluid it has, it also contaminates the another leaf. Means, suppose this is infected leaf pulp. Okay, when this infected leaf pulp is filtered out by Chamberlain paper and its fluid is applied into healthy leaf, this leaf get affected. This Chamberlain paper can filter the bacteria, but again it affected. It means this disease is not caused by the bacteria, because if that was caused by the bacteria, it will not filter out through Chamberlain paper. It means some microorganisms are such type which is rather lesser or has the least size than the bacteria, and that was termed as the virus. So this experiment was done by the Ivanovsky, and then after Martinus Vezere, he worked on it. Martinus Vezere, he worked on the viruses and said contagium vivum fluidum. Contagium vivum fluidum. But it means that this is the contaminated vivum, that is the poisonous. The poisonous fluid which is contaminated, that cause the disease, and then he was given the term virus, and He says that these contaminant that is the virus, cannot be cultured. But after that, Parker and Nye, Parker and Nye, as I told, the prior research are discarded by the new research. And Wesley says virus cannot be cultured in artificial medium, but Parker and Nye, they culture the virus in laboratory. In laboratory. It means virus can be cultured. Then after J. Endler, he came and he cultured the polio virus on corneal corneal membrane of the birds. Now they came to know that virus can be cultured. After that, what they say, what is virus? They got some new information regarding them, and then they got that virus is made up of nucleoproteins. Virus is made up of nucleoproteins. Suppose this is a virus. What is this? This is envelope, right? This is protein code, and this is nucleic acid. So, virus is a nucleoproteinaceous particle or nucleoprotein particle which is dead outside the living cell. What is envelope? Envelope is made up of some proteins and its unit is known as tapenomere. Tapenomere. Okay. The protein code, it is termed as capsid. Capsid. And its unit is termed as capsomere, right? That is the capsomere and the nucleic acid. There are two types of the nucleic acids are present. Either DNA or RNA, but never DNA and RNA. Are you getting it? It means nucleic acid present, either they have DNA or RNA, but never DNA and RNA both, as in the prokaryotes and eukaryotes. Okay, so either it will has the DNA or it will have the RNA. This DNA and RNA may have the part, they may be single stranded, point and circular DNA, they may be double stranded. So different examples are there. Actually, these viruses are obligate parasites. Obligate low parasite. What it means? There are certain viruses which affect the plant and particularly a plant species, whereas the another they will affect the another plant species. The virus some affect the animals, different species. Suppose TMD, tobacco mosaic virus. This virus will affect only tobacco, not other plants. Okay, it means this virus is dangerous for TMD, but not the other. Like we are nowadays, we are suffering from the COVID-19 virus. This virus is obligate parasite in animals, especially human being. Okay, they will not create any effect in the plants. So this is mean of the obligate, means virus affect a particular species of organism, not others. What do you mean by the endoparasite? Endoparasite means they are living inside the organism as a parasite. When they enter in the cell, they become the part of cellular nucleic acid, and then they use the machinery, proteinaceous or enzymatic machinery of the host cell and multiply them and thus spread in numbers, right? So this spreading in numbers, they may take sometimes 1 days, 2 days, 3 days, 4, 5, 10, 14 days. That's why, and this time period is known as the incubation period. Incubation period, right? So as COVID-19, what is the incubation period of this COVID-19? Approximately 14 days. That's why if the COVID-19 virus is entered today, it will multiply inside the body till 14 days, and upon they get sufficient amount, then they will appear their symptoms. So this 14 days is incubation period, then after the disease period, right? So the next thing, as we know, that virus has two parts. One proteinaceous part, and the next one is nucleic acid part. So the nucleic acid part, this complete is virus. They may be virulent, means disease causing, and non-virulent means they will not cause the disease. So the virulent, complete viral particles with virulent nature is called virion. Virion, right? And some parts, the infectious pathogen completely as only nucleic acids like circular single stranded RNA. This only nucleic acid part, they are termed as viroids. So if virus has a complete part, proteinaceous part as well as nucleic acid part, then they will term as virion. And if they have only nucleic acid part, the violent nucleic acid part, then they are termed as the viroids. Some another term, that is the prions. They are the proteinaceous part, means misfolded protein. You will not mess with the prions and virions. It is separate, and this prions is a misfolded proteins which helps to misfold the another same type of proteins. That are the prions. So it is not, you will not mess with the virions and prions. The next thing, as I told, this is the envelope. Only animal virus has envelope. Means virus are of two types. Non-enveloped. Okay, sorry, it is not clear. Okay, I'm writing here. Non-enveloped and envelope. Means if virus particle has envelope, then they are the enveloped virus. And example of enveloped virus is animal virus. But certain virus, they do not have the envelope. They have only protein code that is captured. And the example of non-enveloped virus is plant virus. Oh, here are some points. Now, as we know that nucleic acid, either they have DNA or RNA. Majority, but not all. Always remember it. When we are saying a certain terms in biology, they have its particular meanings because biology is the science of living. You cannot say this is the exact thing because there may be any exceptions are already existing in the world. Okay, generally, if we are saying majority DNA virus are animal virus and majority RNA virus are plant virus. Okay, example of majority DNA virus are animal virus, means animal virus has genetic material in DNA form. Plant virus has genetic material in RNA form. Commonly it is, but they have the exceptions. Okay. The next classification of viruses. Actually, viruses are classified according to its presence or host. As if virus affect the bacteria, then they are termed as bacteriophore. For this phagus, this eating, and bacterium, it is related to the bacteria. So if they infect the bacteria, then they are termed as the bacteriophores. If they infect the cyanobacteria, then they will come as cyanophores, right? And if they will infect the fungus, then as you know, mycology is related to the fungus. So mycophores. Cyanophores infect the blue green algae, mycophores infect the fungi. You remember I told that viruses are obligate parasite. If virus infect the bacteria, the same virus will not infect the blue green algae or cyanophores. Some more points. Actually, virus has the capsid, its unit is capsometer. They have a fixed number. We can obtain the number of capsomeres present in any capsid, and by this number of capsomeres, we can know that what are type of these viruses and they will affect to whom. I'm just giving a formula to you. Number of capsomere equal to 10 into n minus 1 plus 2. Is it clear? What is 10? Number of capsomere present. N is the number of capsomere present. Okay. Now, suppose any virus has two capsomeres, right? We are seeing the age of virus. Suppose 10 to what we found 10 into 2 minus 1 power plus 2, that is 10 into 1 to the power plus 2. It means 10 into 1 plus 2. 10 plus 2, that is 12. Is it? So there are 12 number of capsomeres present, and this is present in virus. This is the example of psi times 174 virus. Similarly, tobacco virus has 42 capsomeres and so on. So they can identify the number of capsomeres present in any virus. There are some points regarding the viruses, why they are non-living. As I told, some virus has reverse transcriptase enzymes, such as RSV virus and HIV virus. Okay, so due to this reverse transcriptase enzyme, they may change RNA into DNA and again from DNA to RNA. That's why the medicines of HIV virus are not easy to make. So earlier the scientist says that virus do not has enzyme systems. Here we are getting that some viruses has some enzymes related to its own function, and the rest of enzymes they will hire from the host cell. Okay. Now the next point regarding the lichens. Lichens. You have studied the lichens in class 9, and they are the indicator of sulfur dioxide. Means if sulfur is present in air, these lichens will not grow. And this happens in the industrial areas. What are the lichens? Lichens are the symbionts. Symbiont means they are living symbiotically. So, who are living symbiotically? Means algae and fungi. What do you mean by the symbiotic relationship? Means they mutual get benefited by each other. Means algae benefited the fungi and fungi get benefited by algae. This is also known as master and slave relationship. Means one will be the master and next one will be the slave. So who will be the master? The organism who are providing the space to live someone. Suppose you, if you are living in a room in hostel, okay, and if you are the paying money for it, the another state who do not have the money and he want to live with you says, okay, you will live with me and you will not pay any money or room rent. I will pay the room rent, but you have to do one thing. What? You will cook the food and cleans the utensils. He says, okay, I will do it. So here is the mutual relationship. You don't know how to get the food, how to cook the food. He is cooking the food, right? He lack the money, but you have the money, so you are giving him space to live. So here is the mutual relationship, right? So algae make the food, and that food is taken by the algae as well as fungi. And fungi because these algae are the planktons and they are just swimming from the water. So they should be have a particular place. So fungi trap the algae and provide the space to live within it. Okay, so fungi provide the space or habitat for the algae, and the algae make the food, and by this food benefited to both algae as well as fungi. This algae, botanically, it is known as the phyco. So, if the phyco have the symbiotic relation, they are told as the phyco biome. Fungi, it is termed as mycobiome. So, algae is phycobiont and fungi is mycobiont. They both are get benefited and symbiotically they are termed as lichens. So today this chapter has been completed. Next day we will cover the next lesson, next topic. I hope you will understand. If you have any query, you may ask in the group. Thank you. Have a nice day.